Hello, and thank you for attending today's webinar. I'm Bridget Mallory, an application specialist here at Nanotemper Technologies. This seminar will focus on a case study in the field of targeted protein degradation, where our Dianthus system was used to quantify both binary and ternary interactions. We hope you enjoy it. In order to have a thorough picture of our degraders, we need a toolbox with structural, biochemical, and biophysical methods. Each one provides a new piece of information to shape our understanding of a degrader's behavior. For example, crystallography data was used for ternary structure characterization, and in vivo degradation assays were used for functionality assessment. And to tie all of this together, this structure-function relationship, we use biophysical assays to quantify the strength of our molecular interactions. In this study, our Dianthus platform was used for biophysical characterization of a set of WDR5 degraders to complement the structural and biochemical characterization previously done by our collaborators from the Structural Genomics Consortium, or the SGC, at the Goethe University in Frankfurt. Our target of interest, WDR5, is a protein that plays a critical role in the process of histone methylation, which is important for the regulation of gene expression. Dysregulation of WDR5 expression and function has been implicated in the development of cancers, particularly through its interaction with the MYC oncogene. WDR5 inhibition has been proposed as a potential therapeutic strategy for cancer treatment, and shown here, you can actually see a crystal structure of WDR5 that was co-crystallized with an inhibitor. The limitation in applying the small molecule inhibitor approach is that WDR5 with one active site blocked can still participate in a number of different protein-protein interactions. To combat this issue, our collaborators at the SGC developed a protein degradation strategy. In a moment, I will detail their biochemical and structural evaluation of 5 different degraders that target WDR5 via the recruitment of VHL. Using our own biophysical methods, we collaborated with the team at SGC by assessing binary and ternary binding affinities. And then we took this data in the context of the degradation assays that were measured in vivo to understand how the strength of those interactions may be impactful on the degrader efficiency. Before getting into the details of the results of this study, I wanted to spend a couple minutes talking about targeted protein degradation, which has emerged as a method to target molecules that would otherwise be classified as undruggable. The process starts with a hetero-bifunctional molecule or degrader molecule that is often referred to as a PROTAC, which can simultaneously bind a target of interest and an E3 ubiquitin ligase complex, forming the ternary complex that you can see at the top of this cycle up here. This leads to the ubiquitination and degradation of the target of interest via the cell's own protein degradation pathway, known as the ubiquitin proteasome system, or the UPS. The UPS consists of a multiple enzymatic step process with sequential action of ubiquitin activating enzymes E1, ubiquitin conjugating enzymes E2, and ubiquitin ligases E3. You can envision that complex up here. The addition of a polyubiquitin chain to the target of interest serves as a recognition marker for its proteolytic degradation through the proteasome. By taking advantage of this machinery, PROTACs can drive the knockdown of a large variety of proteins, including nuclear receptors, transcription factors, skeleton proteins, enzymes, and regulatory proteins, which are dysregulated in cancer and other diseases. Let's take a close look at the degrader structure that is going to be relevant for today's study. Our degrader can be viewed in three separate parts. First, to the left, we have the VHL ligand, which will bind our ligase. To the right, we have the warhead that will bind to our target of interest, WDR5. And lastly, we have this linker region indicated by the dashed line box here. And in addition to the C4 linker that is shown in this structure, you can see over on the right-hand side with our linker library, we have 4 other linkers that can be implemented into this structure. One of the key takeaways from the SGC study was that the linker structure can impact the degrader function. So our collaborators sent us these 5 complexes after concluding that they were the top candidates for biological activity. In addition to the impact that these linkers had on degrader function, crystallography data indicated that the linker structure had a fairly significant effect on the structure of VHL. This graphic here provides an overlay view of multiple ternary crystal structures. And of course, this is a very important piece of information when assessing the structure-function relationship of this complex. Next, I will get into the in vivo degradation assay and show some of the results. The biological activity for each degrader was tested using an in vivo depletion assay known as the HYBID assay. In the cellular-based assay, the HYBID probe is expressed on the target of interest, which in our case resulted in the WDR5 HYBID complex. And then the cells are dosed with the degrader molecule, and after 24 hours, the amount of remaining protein is assessed by a luminescence assay. What you're looking for in the graphed data is that as you add more degrader to your cells, there's a decrease in the luminescence, indicating degradation of the target of interest. The parameters derived from this assay are the DC50 and the Dmax, where DC50 is a value indicating the concentration of your degrader where you reach 50% degradation activity. And the Dmax is referring to the maximum fraction of degraded target of interest. Looking at the values on the right-hand side in the table, what we take away here is that the C4 degrader showed the most depletion in the assay, while C3 and C5 showed less depletion. The aryl complex, or compound, appeared to have a similar DC50 value to C4, but it did not show the same amount of degradation. And finally, the PEG complex was concluded to be inactive in vivo. Interestingly, all of our degraders, of course, with the exception of that inactive PEG compound, displayed the characteristic hook effect inflection, which leads to decreased activity at higher concentrations of degrader. For the remainder of this presentation, I will dive into the biophysical characterization of both the binary and ternary interactions that we measured using our Spectral Shift technology. But before that, it's important to note that in the SGC publication, isothermal calorimetry, or ITC, was used to determine the dissociation constants, or KD, for these interactions. And while ITC is referred to as the gold standard for thermodynamic characterization of interactions in solution, it does require a large amount of material, and it's very labor and time intensive due to the fact that it can be relatively difficult to optimize. More importantly, the deconvolution of binary and ternary interactions becomes very difficult with ITC. Knowing that our target of interest, WDR5, is a somewhat tricky protein to work with, and that the degrader molecules were produced in a pretty low yield and are rather insoluble, we wanted to see if we could provide the SGC with some more biophysical data. So we embarked on this collaboration aiming to characterize the ternary complex formation using our Dianthus platform with our new Spectral Shift technology. Our Dianthus system provides high throughput for measuring binding affinities completely in solution, where it uses the fluorescence of a small red dye for detecting binding events. The samples are measured in 384 well plate, which can be integrated directly into your existing workflow using a liquid handling system. To give an idea about the sample consumption, each well requires a total sample volume of about 20 microliters with target molecule concentrations around 20 nanomolar, sometimes less. Before showing you some of the Dianthus data, I would like to talk about the use of the red dye for the fluorescence-based measurement. Using WDR5 as the example here, we used our nanotemper red NHS labeling kit to covalently label the protein at the free lysine residues. And the way that we quality check our protein before and after labeling is by using nano-DSF to assess that the thermal stability has not been significantly negatively impacted after labeling. Since nano-DSF is a technique that utilizes the protein's intrinsic fluorescence for thermal unfolding profiles, the red dye is not going to interfere in this measurement. The data that you're looking at here are two thermal unfolding profiles for WDR5 before and after labeling. And what we saw was a fairly typical temperature shift after the red dye was attached to WDR5. And with this data, our team concluded that the slight change in the melting temperature was satisfactory evidence for very little structural change with the presence of the red dye. I'd like to also take this time to draw your attention to this residue, lysine 237, which we know from our crystal structure is in very close proximity to the binding site that is involved in our ternary complex formation. I point this out now so that you can keep this in mind when we're viewing the binding data, where you'll see that even when you have a potentially labeled lysine residue located so close to the binding site, we don't see any evidence for binding site interference. Now let's talk about the experimental design. Our first step was to assess binary binding affinities. So in order to fully characterize the ternary complex formation, we start by looking at the interaction of the degrader molecule with its binding partners individually. And to maximize the throughput of our Dianthus, we divided this 384 well plate into two halves with VHL interactions on the top and WDR5 interactions on the bottom. Please note that with our VHL interactions on the top half, VHL was labeled with our nanotemper red malamide dye kit, which covalently labels free cysteine residues. And as previously described in the WDR5 interactions, WDR5 was labeled with the red NHS kit to label free lysine residues. We designed this plate so that each degrader molecule was tested in duplicate 16-point binding affinity assays, with an additional assay you can see here for negative control against each of the proteins. With our pre-planned plate design and a liquid handler such as the Integra Assist robot that we use in our labs, and our Spectral Shift technology, this entire workflow can take less than 2 hours. The Dianthus itself is capable of measuring this full 384 well plate in less than 35 minutes when Spectral Shift is used. So now let's take a look at the binary binding results. Our binding assays with VHL, shown on the left-hand side, resulted in beautiful isotherms, all with KDs that we would expect for this interaction. The binding assays for WDR5, shown on the right-hand side, also resulted in very nice isotherms, but we see this rather interesting effect here where the scale of the isotherms are quite different from one another. But this should not be interpreted as insufficient binding. Rather, this gives us some indication that spectral shift not only provides biophysical data in the form of KDs, but also a hint at the fact that there may be structural differences between the degrader WDR5 complexes. Moving on to the numerical KD values, you can see in this table down here that we were able to determine a KD for each of the 10 interactions that we tested. We were able to compare the WDR5 data with the results that we had made available to us by the ITC data. And in all cases, those KDs were quite comparable to each other, with one exception being the PEG linker degrader molecule, for which the binding affinity to WDR5 could not be determined with ITC. And I'd like to also point out that all of these KDs measured by spectral shift, when including optimization steps all the way to protein labeling and assay development, took only a matter of days to complete, where the ITC results took weeks to optimize and measure. And now, finally, we can move into the results for ternary complex formation, which is where we'll see that the Dianthus really thrives. So let's take a close look at one data set before looking at the results for all 5 linkers. First, what we have here is the binary binding affinity data for the VHL degrader interaction. Next, we see a new isotherm where the same labeled molecule, VHL, is measured when there is unlabeled WDR5 present in 10-fold molar excess. I will point out in this data set 2 very important components for characterizing the ternary complex formation. First, we have evidence of the hook effect, which is indicated by this arrow here. And second is this alpha value that indicates a large shift in the KD due to cooperative binding activity. So for the big reveal here, I have all of the ternary binding biophysical assays presented all at once for each of the 5 degraders. You can see that for all 5 degraders, spectral shift results revealed or observed a positive cooperativity and detectable hook effect, indicated by this arrow here. And next, I will show you the numerical results and put them back into the context of the SGC study. In this table on the left, I present some of the important quantities that were derived from those biophysical assays. You can see in the first column, we have measured KDs for the degrader VHL binary interaction, followed by the KDs for the same interaction when WDR5 was present, forming that ternary complex. And lastly, we have the cooperativity factor alpha, which can be defined as the binary KD value divided by the ternary KD value. In the graph on the right, we provide a visualization of a potential correlation between that degrader activity that was measured in vivo with the dissociation constant KD. And while our study did not include enough degrader molecules to draw very final conclusions from this, the correlation supports an emerging evidence in the field of targeted protein degradation that Biophysical data could be used to screen degraders in vitro, before moving into cellular-based assays. And these cellular-based assays can be very costly and time and labor intensive, so it is nice to screen these molecules beforehand. Our spectral shift assays can provide the KDs for each binary interaction, as well as the KD for the ternary interaction, plus that cooperativity value alpha, as well as the degrader concentration where we see an onset of the hook effect. Taken all together, these parameters could be used to rank degrader molecules for their biological potential. And to conclude, I will leave you with the following take-home messages. So first and foremost, we completed this biophysical evaluation of 15 different interactions, where 5 of those were ternary complex formations, in days rather than weeks. That included the complete optimization for WDR5 to find suitable buffer conditions for both labeling and running our assays. And we accomplished this study regardless of the limitations that were presented by relatively insoluble degrader molecules that were produced in rather low yield. Since we operated with an intelligently designed experiment, we were set up for success with high throughput to get good quality results quickly. And again, as a reminder, the results were 15 KDs after about 3 days of work. And finally, this work supports that emerging hypothesis that degradation activity in vivo can be correlated to biophysical data, which will hopefully allow for biophysical evaluation to optimize candidates before moving to cellular-based assays. And now I'd just like to thank our collaborators again, because none of this work would have been possible without the efforts of the scientists listed here. From the SGC, this was the hard work of Anya Dola, Andreas Kramer, Stefan Knapp, and Bikash Adhikari. And from Nanotemper, this was the hard work of my colleagues, Nate Adams and Andreas Longa.